Okay, could somebody confirm you can hear me?
Yes.
Yep.
Thank you. All right, before we begin, are there any questions? All right, if there's no questions, I have done some grading. I have most people's lab 2 graded, and I hope to finish the rest of the people's lab 2 tonight, probably during the lab session tonight. Um, I'll talk more about the lab in the lab. And I also got the worksheet for the previous lecture graded. Were there any questions on the grading or anything like that? All right. We had talked about phospholipids last time, and we mentioned that they are a biphasic molecule with the phosphate portion of the phospholipid being uh, hydrophilic, meaning it likes to dissolve in water. And then the tail portion of the phospholipid, the fatty acid portion, is hydrophobic and it likes to be away from water. Phospholipids are the main molecule in the cell membrane. When you get a bunch of phospholipid molecules and you put them together in water, they will start lining up like this. Let me make this a slideshow. Well, they'll start lining up like this so that the fatty acid portions of the molecule will come together and be away from water, which is on the outside. And then the fatty acid portions will be together and the heads will face the water and it'll just form a circle like that with the water being outside and the heads being on the outside of the phospholipids, the fatty acid tails all being on the inside. And then there will be a layer of phospholipids with the head facing outward. And the inside of this would be, of course, water inside here. And this should remind you of something. What does that look like to you? Anyone?
Sunflower or lemon?
Okay, let's go to microbiology. This should remind you of a cell. The cell will, especially an animal cell, will be surrounded by the cell membrane. And that's what this is here, with phospholipids being on the outside of the membrane, and I mean the phosphate head being on the outside of the membrane, and the phosphate heads being on the inside of the membrane, with the tails making the phospholipid bilayer of the cell membrane. And then the outside of the cell, if we're talking about in blood, will be mostly water. And it doesn't really matter where the cell is because most of the body is water. So wherever the cell is, mostly it's going to be water outside the cell. And then mostly water inside the cell. And that will be the cytoplasm inside the cell. So this should remind you of a cell. And as I stated, the cell membrane is mostly composed of phospholipids, with the phosphate portion being the outside and on the inside, and then the fatty acid portions being inside the cell membrane. All right, any question about that? If not, let's talk about another group of lipids that we call steroids. If you remember, there were three types of lipids: simple lipids, complex lipids, or fats and triglycerides. And well, actually, there's four because there's phospholipids, that's different. And then the other group is the steroids. A steroid is four interconnected rigid carbon rings characterizing this group. And there's a 1, 2, 3, 4 carbon rings. If the steroid has an OH group attached right there, we then call it a sterol. Sterols are an important part of the animal plasma membrane, where we have cholesterol, which is a sterol. In fact, that's shown here. It's inside the cell membrane. So we do have phospholipid molecules in our membrane, and it is the main molecule, but we also have cholesterol. And the purpose for cholesterol is to give strength to the cell membrane. If you remember, animal cells don't have a cell wall, and so they have cholesterol in their cell membranes to give them a little more strength. Cholesterol is only found in animal tissue, meaning in animal cells. It is not found in plant cells. Any question about any of that? Steroids are a lipid which is not made up of a monomer. A steroid is one molecule, like cholesterol right here, and it has no monomers. You wouldn't call it a polymer either. It's a large molecule, but it's not a polymer because it's not made up of smaller subunits. All right, any question about lipids? If not, let's move on to proteins. Proteins are essential in the cell structure and function of both a cell and an organism. Proteins differ. They may be structural proteins found in bone, muscle, or hair. They may be enzymes which speed up chemical reactions. They may be transport proteins that move chemicals across the cell membrane or across a organelle membrane. They may be regulatory proteins that regulate a body function, like a hormone. Like in children, they have a growth hormone. Proteins may be responsible for movement, and it's true that we have proteins in our arms and legs for moving, but you also have proteins for cellular movement, such as the protein that makes up flagella. Proteins can be responsible for some bacterial toxins. So the protein itself is toxic. And then proteins can be antibodies, proteins that help defend an organism against an infection by a microbe. So this tells you that proteins have a bunch of different functions. Proteins are more diverse than any of the other four biological molecules. They are more diverse than the other biological molecules because they aren't really necessarily larger, but they have more building blocks, like complex carbohydrates only have a handful of building blocks, such as glucose, fructose, and galactose. And fats like triglyceride or lipids only have a few building blocks, and some of them, like cholesterol, have no building blocks. Cholesterol is just one molecule. But like a phospholipid has three building blocks, a fatty acid, a glycerol, and a phosphate group. See, carbohydrates have only the simple sugars, which are a handful of building blocks. Oh, I was trying to remember the other group. The nucleic acids only have four building blocks, the different four nucleotides. But proteins have a building block that we call an amino acid, and there are 20 different amino acids, at least in humans, and in most organisms, they have 20 different amino acids. So proteins consist of the monomer that we call amino acids. This is the generalized shape of an amino acid, where it has an amino group on one end, or carboxyl group on the other end. Between them, they have the central carbon. Hydrogen is connected to that central carbon. And then on the fourth position of the carbon, we have an R group. The R group can vary. R stands for, well, essentially anything we want it to, but in amino acids, it's the R group that varies that makes the different amino acid. And then you just link these amino acids together to form a string of them to make a protein. Here we're looking at one R group for tyrosine. Here are some of the different R groups. You don't need to know these R groups. I'm just trying to show you that each amino acid will have a different R group. So there are 20 different amino acids. That means there are 20 different, at least in humans, there's 20 different amino acids. So there will be 20 different R groups, one for each amino acid. The simplest R group is simply hydrogen, which we see on glycine right here. Your R group can be, it can be nonpolar, like a methyl group here, a branch methyl group here, a branch chain of methyl groups there. These are all nonpolar R groups. But the R groups don't have to be all nonpolar. Some R groups are, or at least have regions that are polar. Like this R group here has this OH group, and that's polar. The rest of the R group is nonpolar. In this R group, wherever there's the NH, it is polar. And then this region of the R group would be nonpolar. Better shown on this one. So this would be a polar region of the R group, and then the rest of the R group would be nonpolar. Amino acids always have a polar group. Actually, they have two polar groups because the amino group is always polar. The carboxyl group is always polar. And then the R group can be either nonpolar like that one, or it can have polar regions like this one. Any question about that? Okay, amino acids are linked together by a peptide bond. A peptide bond is formed by dehydration synthesis. You remove the OH from this amino acid, the hydrogen from that amino acid, and then that combines the nitrogen with this carbon here, which is an unusual unusual bond between nitrogen and carbon. That's not only unusual in living organisms, it's also unusual in the universe. We don't often have a bond between nitrogen and carbon. We do have it in amino acids and proteins when they the amino acids link together. And so we call this bond a peptide bond because it is found in peptides. And if you remember, a peptide is a shorter version of a protein. When you're adding amino acids together, we call it a peptide until you add the last amino acid on, in which case we then call it a protein. So peptide bonds are formed by dehydration synthesis, and we link the amino acids together to make a polypeptide chain. And when the last amino acid is joined to the growing protein, we then call it a protein. But it's not a protein until the last amino acid is added. You can call it a growing protein, but not a protein. There are 4 levels to the protein structure: a primary level, a secondary level, a tertiary structure or level, and then a quaternary level or structure. The primary, well, let me go ahead and show you the movie.
The hardest part about a writing assignment is getting started. I'll use Grammarly to illustrate how AI can be your...
In this
Okay,
video, we're going
commercial.
to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost 2 once we lost water. So this is a dipeptide because we have 2 amino acids combined. And so this amide bond that we see here, that is the peptide bond. we formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein, and there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid, and each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions about that video? So let's talk about the 4 levels of protein structure. The primary structure is the specific sequence of amino acids in a polypeptide chain. So when the protein is made, it will simply be a sequence of amino acids, one after the other. And that's the primary structure. Any question about that? Now, when the protein is first made, it's just a linear sequence of amino acids. However, the secondary structure soon forms as the amino acids of the protein start interacting with each other. And initially, it's only localized interactions where neighboring amino acids interact with each other. And that we call the secondary structure. There are different types of secondary structure. The video told you there are 2 you need to talk about. There's actually 3. You don't really need to know these, but you can have the alpha helix, which is a shape like that or like that. The beta pleated sheet, which is a shape like this. And then there's the random coil, and let me just go ahead and draw out the random coil. Try and find paint. The random coil is where the amino acid the protein just randomly takes on a shape. Let's make that come out a little further. Something like that.